there was two scientists who were responsible for disproving spontaneous generation, or proving that spontaneous generation was incorrect. The first being Francisco Redi, who did what we call the fly meat maggot experiment. And the second we'll talk about next, who was Louis Pasteur. So Francisco Redi, his main objective was to disprove that spontaneous generation was incorrect, that there was no way that something non-living could release any type of force to produce a living organism. So what he did is he took 6 different beakers and he put a piece of meat in each one of these beakers. 2 of the beakers were left exposed to the open air, so air, microorganisms, insects could all be exposed to the meat. The other 2 were closed with an airtight lid, so nothing would be exposed to the meat, no air, bacteria, insects, nothing like that. And the last 2 beakers were covered in a gauze wrap, so air could freely flow through, but larger things like insects would not be able to get through. They would settle on the gauze. So if spontaneous generation was correct, if something non-living, such as a piece of meat, was able to produce a living organism, such as a fly, then one would assume that every single one of these pieces of meat would have flies on it, because it would have been the meat that produced the living organism, which was the fly. But the only pieces of meat that actually had flies growing on them was the one where the meat was actually exposed to insects or flies. So what happened was flies laid eggs on the meat, which eventually turned to maggots, which eventually turned to more flies. The other containers where the meat was not directly exposed to the flies did not have flies on the meat. And again, that was because flies produced flies, so only the meat that had exposure directly to the flies would have had more flies present on it. Meat is a non-living organism. It doesn't have the ability to produce a living organism such as an insect. So Redi was the first person who came in and said, okay, spontaneous generation is not correct. Unfortunately, not all people believed in him, so a second scientist had to come in and disprove spontaneous generation, which was Louis Pasteur. So Louis Pasteur, he disproved spontaneous generation by using something called a swan neck flask. So his big contribution was this swan neck flask, which we can see here. It's just a standard flask, but the neck of it is curved like a swan's neck. And what he did was put sterile broth, so broth that was not contaminated with any type of microorganisms, into the swan neck flask. And he just left it sitting there, and he said, okay, if this broth is able to produce a living organism, like bacteria, then the broth will get cloudy. That was an indication that bacteria were growing in the broth. So when he left the swan neck flask sitting out with the clear, sterile fluid in it, it always stayed clear and it always stayed sterile because it had no ability to produce microorganisms. Even if microorganisms like bacteria would get in through the air, they would settle here in this region and would not be able to get into the flask because of gravity. Now what he did find is if he broke off the neck of it or tilted it so that the broth was allowed to go into this curvature part, then the solution would get cloudy and would get contaminated with bacteria. But that's because bacteria were able to get in and produce more bacteria. So bacteria produce bacteria, not the broth. The broth was a non-living substance. It had no ability to produce a living organism like bacteria. However, when the broth was exposed to bacteria, the bacteria would grow inside the broth and produce more bacteria, and that's what would cause the broth to get cloudy. So this was the final kind of end of spontaneous generation and that belief that something non-living could produce a living organism. So now we're consistent with biogenesis and the idea that living things produce other living things of the same kind. Bacteria produce bacteria, flies produce flies. and we've officially disproved spontaneous generation. So other big contributors to microbiology, one being Anton von Leeuwenhoek. He is considered the father of microbiology because he was the first individual to develop microscope lenses that were magnified enough to see microorganisms, or things like fungi, protozoa, bacteria, those sorts of things. He actually didn't have a background in microbiology specifically. He was a linen merchant and had developed these lenses to do thread counts, but then at some point in his career got tired of looking at linen and started to look at pond water and dental plaque scrapings from individuals' mouths. And what he found, he called animalcules, which were these little tiny animals. We now know those were things like protozoa and fungi and bacteria, but those terms didn't exist at that point. So he called them animalcules, and for this reason, for him establishing these lenses that could visualize animalcules or microorganisms, he is considered the father of microbiology. Another father within the realm of microbiology is Joseph Lister. Lister is considered the father of aseptic surgical techniques. It sounds absurd, but at one point in history, it was not believed that microorganisms could be transmitted from person to person. So it was completely normal for a doctor to be with a patient who had bubonic plague and then go into the next room and deliver a baby without so much as washing their hands. And what Lister observed was that it was safer for mothers to deliver their babies at home than it was for them to deliver babies in the hospital, where they'd be more prone to diseases. So he was the first to kind of consider that diseases could be transmitted, and what he did was start to develop aseptic techniques that would reduce transmission. Simple things like soaking surgical instruments, wearing gloves, washing hands, all those things that we kind of take for granted nowadays as being common sense, were founded under Joseph Lister. And once those practices were started to be used in a hospital setting, it cut down the rate of infectious diseases substantially, which makes obvious sense. So for that reason, he is the father of aseptic surgical techniques. A simple way to remember him is Listerine was actually named after him. Listerine is an aseptic mouthwash. Joseph Lister is the father of aseptic surgical techniques. Another big player in microbiology was Edward Jenner. Edward Jenner is famous for creating the first immunization, which is the first vaccine. He created the vaccine towards a disease that is known as smallpox, which is something that we don't have in the natural environment anymore. It has been eradicated, but at one point in history, it was a very deadly viral disease. And he noticed that while most individuals were getting smallpox, and a large portion of those were dying from the disease, milkmaids were not getting smallpox, and everyone else was. And he made the connection that milkmaids would get a disease called cowpox, which is similar to chickenpox in the sense that it's contagious but not deadly. Typically, you just get some itchy lesions on your skin, and you make complete recovery within a few weeks. He would notice that they would get cowpox but not get smallpox. So his assumption was, if you get cowpox first, you must be unable to get smallpox. And we now know that the immunity that you get from the cowpox disease has a protective response that makes you immune towards smallpox. But he just said, get cowpox first, don't get smallpox. So what he did was he took fluid from the cowpox lesions that would have had the cowpox virus and injected those into individuals, and that would elicit a response, a memory response, that would protect individuals from exposure to cowpox again, but also exposure to smallpox. And that was kind of how they created the first vaccine. So Edward Jenner is responsible for the first vaccine, which was towards the disease that we know as smallpox. Paul Ehrlich is famous for discovering something called the magic bullet theory, which was basically this idea that medications could be designed to target the pathogen and cause minimal damage to the host. So anytime you take any type of medication, such as, for example, an antibiotic, you want it to go in and get rid of the infection and not cause a lot of different side effects. During his time, what he was trying to treat specifically was syphilis, which is a sexually transmitted disease, but it can spread to the brain and it can become very deadly. And during this time, a lot of people were succumbing to syphilis. And what they used to treat syphilis at the time was an arsenic compound, which arsenic would kill the microorganism that causes syphilis, but it was very toxic to the host. So in the process of treating the infection, there was a lot of really bad side effects. Some could even be lethal. So he said there has to be a better medication. There has to be a medication that acts like a magic bullet with precision and goes in and gets rid of the infection without causing all these different side effects. So he eventually did develop a medication that could cure syphilis without all the side effects as arsenic compounds. But because of that, he is responsible for terming what we call the magic bullet theory. Robert Koch did a variety of things within micro, but one thing he's most famous for are developing what we call Koch postulates, which are basically a series of steps that allow us to identify the causative agent of a disease. I don't need you to memorize these steps, but I want you to understand what these steps do. If we have a new disease and we want to say that this microorganism causes this disease, it's not enough just to say it. We have to go through these steps, and if we can confirm all these steps, then we can identify or apply causation and say, okay, that microorganism actually causes that disease. So the first step, the specific causative agent must be found in every case of the disease. If we want to say that this new type of bacteria causes this new type of food poisoning, well, every single person with that new food poisoning better be infected with that specific type of bacteria. The next thing, the disease-causing organism must be isolated in pure culture. So if everyone has that case of food poisoning and you want to say that this new strain of Salmonella caused it, you're going to need to collect a sample from the patients, which in this case would probably be a stool sample, and you're going to need to grow that strain of Salmonella in pure culture, meaning it would be the only bacteria present in the petri dish. Then what you're going to do is step 3. You're going to inoculate the sample into a healthy animal who must then possess the same disease. So you would take the strain of Salmonella, inject it, or transmit it to a healthy animal, and that exposure would result in that animal then getting the same infectious disease in that case. And then the last step, you would then collect a sample from the infected animal and grow the specific strain in pure culture again. And if all those steps, if you're able to validate through all those steps, then you will be able to definitively say, okay, this specific microorganism is able to cause this disease. So what these steps really do is allow us to apply causation. They allow us to say, okay, this is the causative agent of this specific disease.